Okay, welcome back to ZOO3649 Evolutionary Genetics 2020. You're with me, Prof. Moodley, and this is lecture 5. And as I said last time, in the last lecture, we are going to go over this again. This is refreshing our memories from ZOO2544 last year. You will need, and we're refreshing this. Why? Because it's important. We need, when we're doing genetics, especially when we're looking at populations and conservation and zoology and botany, even in micro, we need to know how to measure the variation. We need to know how to measure it. Okay? And if we don't know how to measure genetic variation, we then do not use the genetic data for what we can use it. So why do we generate the genetic data? All that genetic data we have been generating with NGS and all these new technologies, now we are analyzing that data. We figured out how to store it, how to access it in our bioinformatics. Now we want to do some basic analysis on that data. Okay? We want to figure out how much variation is there. So there are lots of different ways to measure genetic variation. I'm going to go through the main ways of doing it, and that really depends on how the variation is organized. Okay? Each way of measuring variation is different to the other, and so when we decide which measurement do we want, which way do we want to measure the genetic variation of something, we have to figure out the way that is most appropriate for our purposes. It depends on your study. If you are looking, for example, at a medical study, no? If you are looking at a patient who has cancer, or a patient who has HIV, or a patient who has COVID, and you are trying to measure variation, you will do that at the level of the individual, okay? Because you're trying to figure out if this individual has a disease, or if it does have a disease, how can we treat it? You need to know copy number variation. You need to know individual level variation, okay? But then what happens if you have a game ranch in Limpopo full of wild animals, but your game ranch is fenced, and so your animals are busy breeding there. So how long is it going to take for the diversity to go down? The genetic variation will always go down if it's a closed system. Well, you will learn all about this during 3649 this year. If you close your system, there's no gene flow coming from other populations, so your genetic drift is going to hammer that population and make the genetic variation go less. You have to be able to, how will you measure that variation? You need to measure population genetic variation because it's a group of animals in your ranch. What about if you're a farmer? What if you're a farmer and you have 10 cows? So if you keep your cows by themselves and they're not allowed to mix with other cows, then you will need to bring genetic variation from outside every few generations. You cannot keep mating with the same bull every generation after generation. You cannot have a father breeding with his daughters or granddaughters, okay? Because that will give inbreeding, and we will also deal with inbreeding during 3649. So it's very important to measure genetic variation for so many reasons, as I've just outlined to you, okay? Those are just 3 reasons I gave you, but imagine there are hundreds of other reasons, and you always need to figure out first, what kind of variation do I want to measure? What way is best for what I'm studying here, okay? And so it is important to know then what level can we organize the variation, okay? So we can organize the variation. You can say we can measure variation depending on the genetic marker, okay? That's a very normal way to do it. The other way to do it is we can measure variation by the number of copies of DNA or RNA, so in the genome or transcriptome, that means in the DNA, the genome, or the transcriptome, which is the RNA, how many copies of a particular gene are certain cells, liver, heart, skin, muscle. What is the difference in the number of copies? between these tissues. That can be in a single patient. Okay? So the differences in the number of copies is a way to measure the variation. Okay? The copy number. And then what happens if you want to organize things in a hierarchy? You can look at individual variation, like the number of copies of DNA, or you can look at population. Okay, if you have one population that is closed, is that population joined to another population? Is there gene flow between the populations? And what about the genetic variation of a whole species? So you can measure genetic variation at different levels. Okay, so that's what I mean by the third one, the hierarchical organization. So let's start with the kind of marker used. This is very important, and I think if you understand this, this part here, you're going to understand a lot in this module, and you're also going to understand a lot in postgraduate studies. So please, memorizing is for the people who just want to pass and finish their degrees. If you're going to try and do postgraduate and you're trying to memorize, you're in third year, you don't have a hope of passing honors. Okay, so forget about it. If you're memorizing in third year, you're not going to make it in honors. All right, so that's why I'm encouraging all of you, don't memorize, understand what I'm saying. Okay, don't just memorize the slide. You're not going to get far. You might pass, you also might not. The questions are not going to be asked the way you can memorize. Okay, so you're going to be in trouble either way. You've seen what happened to you in 2544 when you memorized. Half of the class failed. So please don't let that happen again. You don't have to memorize. Okay, you have to get it. All right. So please, what I'm saying now is very important. I'm gonna explain it slowly, but you must take it on board, right? So we are looking at markers for diploids first up. All right. Now, what is a diploid? Diploid is like you or me, fly, a tree. Some trees are even polyploid, okay? But diploid is basically any organism, usually a eukaryote, with sex, basically, okay? So when there's a male and a female, and they mate, and the male gives one allele, and the female gives another allele, like mom and dad, right? So you get one from each parent. So if you've got one copy from each parent, you are a diploid, and your parents were also diploid. Okay, so now with diploid markers, so basically markers that are in the genome of anything with two parents. Now you want to be able to tell what was given by mom and what was given by dad. Okay, that's very important to know. And we are now talking about those markers that allow you to tell what came from mom and what came from dad. And they're called phased markers. They're diploid markers, but they can tell the phase of the two alleles. Okay? They are called phased markers. Okay? They can distinguish between which parent gave which allele. All right? That's important to know. Which allele came from mom, which allele came from dad. So in other words, they are able to tell us which individual is a homozygote. In other words, both parents gave the same allele, right? And which individual is a heterozygote, which means mom gave you one allele and dad gave you another different kind of allele. Okay? Then you're a heterozygote. So phased markers are really important. You cannot, for example, you cannot measure heterozygosity as a way to measure genetic variation if the marker is not phased. Okay? So when you are doing observed heterozygosity, expected heterozygosity, and so on, yeah, the marker has to be phased. You cannot get heterozygosity from an unphased marker. Even if you're looking at a diploid, you have to have a phased marker. Okay, what is an example of a phased marker? An allozyme, microsatellites, RAD-seq, GBS. Okay, you've noticed the last two there are NGS markers. The first two are not. Microsatellites, as you know, are small pieces of repeated DNA. They are amplified through PCR. Allozymes, as you know, they're not actually DNA, they are proteins. And they are proteins with a slightly different isomer, and they run through a starch gel at a different charge. Okay, so this here is a protein gel. Let me get my mouse to work. Okay, here we go. You see there, my mouse is moving. Yeah, this first one, the black and white picture, is a protein gel. And you see each lane. You see there? Each lane, 1, 2, 3, 4, 5, 6, and so on. Each lane is a different individual. The lane was loaded here, right? That is the top of the gel. And the proteins are moving to this side of the gel on the basis of charge. Okay? And they're separated not on the basis of size, but on the basis of charge. Okay? Unlike DNA, which is run through an agarose gel, this is not an agarose gel for proteins, it's a starch gel. And DNA is run through an agarose gel and separated on the basis of size, not charge. Don't get that wrong. Okay, this is a protein gel here. It's an allozyme gel. But what can we tell about these proteins? So, for example, when you have one band, an individual, this one here, he has one band. This one here, it's too difficult to see a second band. Maybe there's one here. This individual definitely has maybe there's two here as well. But this individual here definitely has one. This individual definitely has one. This individual definitely has one. What do you think for this protein? What do you think the guys who have one? Are they heterozygote or they're homozygote? Huh? You got it, right? They are homozygote. Why? Because mom gave this allele, and guess what? Dad also gave the very same one. So they are running on top of each other through the gel. Okay? You can't tell one apart from the other. That's a homozygote. Look at this one right beside him here, to the left of it. This one has two bands. This other one to the left has two bands. The one to the far left also has two bands. What can you tell? Mom gave one and dad gave the other. So that is two bands. You have a heterozygote. So now let's look at this one. This is basically almost the same as looking at a gel. This is now a microsatellite profile, and the microsatellites were amplified with PCR, and the primer was labeled. Okay, so when the PCR products are run through a gel, okay, now they're PCR products, which means they are DNA. They run through an agarose gel or an acrylamide gel. Microsatellites, because you have to separate two base pairs. Microsatellites are repeat elements. There's usually two base pairs, like a CA or a GT or a GA, that are repeated. Okay, so you want to be able to tell two alleles which are two base pairs apart. You need a very, very, very good matrix to separate DNA on that smaller size. So you don't use agarose, you use acrylamide. Okay, I told you we were going to get more details compared to 2544. Okay, these details you have to know. Acrylamide can separate microsatellites, okay, alleles, because they need to be high resolution. So what is happening here? This is run through a gel, which is actually in a tube, and as it comes out the other side, there's a laser to read the label on the PCR product. So we know exactly what size each of these peaks are, and when the laser reads the label, it has a peak. Okay, so here you see these two big peaks. What is that telling you? That's telling you that that individual is a heterozygote. Okay, look at this here. This individual has also two big peaks here. That is also heterozygote. One of this allele here is the very same size as this allele in another individual. So each one here like this, each line 1, 2, 3, 4 is a different individual. It's like a lane 1, 2, 3, 4 being a different individual. So if you like, these two peaks here are telling you that individual is a heterozygote. The same like these two peaks here are telling you that that individual is a heterozygote. But that you are telling heterozygosity using two different markers. But both these markers can tell the phase. Okay? These are phased markers. You can tell what allele came from what parent. Okay? Now look at this one, this microsatellite here. This microsatellite only have one big peak. Why? Because that one is a homozygote. So you see, when you have two big peaks like that, you have a heterozygote. When you have one, you have a homozygote. So it's exactly the same. You see two bands on the gel, you see a heterozygote. One band, you see a homozygote. All right. So what is one other marker that can be phased? No? There's one other marker that can be phased, and that is the last one I've put here. Whole genome sequences that are greater than 16x coverage. Now, people, if you're scratching your head thinking, what does he mean by coverage? Please go and watch the lectures before. Okay? Don't skip lectures because that's why you don't know what coverage is. I explained very well in the previous lectures what coverage is. You better know what it is. You will be examined about that. Okay? And why over 16x coverage? Why does the whole genome sequence have to be over 16x coverage in order for us to be able to phase it? Okay? For it to be phased, it needs to have a high coverage. Why? You are going to be examined on that. There's no doubt about it. Okay? You better know the answer. All right. So, how about just finishing up with phase markers now? Why is there such a big deal about them? Why is Moodley going on and on and on about phased markers? Because they are the most useful markers you can have in genetics. It allows you to do population genetics. You can figure out whether the population is in Hardy-Weinberg. You can in other words, you can figure out if the population is evolving or not evolving. You can figure out what force of evolution is acting on that by studying the allele frequencies in the diploid allele frequencies, both phases, and how they are going through the population. Okay? And so these are very, very useful markers. In fact, they are the best markers you can get in genetics. Phased markers. Okay? So that is why I'm talking about them so much. But there's another kind of marker for diploids. It's called an unphased marker. It's also in diploids. You can also use it in diploids like us and all other kinds of eukaryote diploids. Okay? But it is not phased. In other words, it cannot tell what allele has come from mom and what allele has come from dad. Okay? You are unable to say this is a homozygote or this is a heterozygote. You can't tell. Okay? So you can't get that information, the what is coming from which parent. All right? They are called unphased markers. RFLPs, restriction fragment length polymorphisms, rapids, AFLPs, and Sanger DNA sequences that are done on diploid on diploid markers. Okay, a Sanger sequence of a gene that is in the nuclear genome, in other words, the diploid part of the genome. Well, that is not going to be phased. Okay, and lastly, I've got here an NGS marker. You see that? Whole genome sequencing again. But now this is giving you a clue as to to the question I asked you in the last slide. Why do you need above 16x to be phased? So here you are seeing if it's below 16x, what? It is unphased. You cannot call heterozygotes if coverage is low because you cannot distinguish between a heterozygote and a mistake made by the NGS machine. There are too few sites. There's too few coverage, too low coverage for that particular site. You are unable to tell. But if you have more than 16x coverage for that particular site, then you'll be able to tell what is a homozygote, what is a heterozygote, and what is a mistake. Okay? Because you've got that site represented at least 16 times. All right. So these are unphased markers. Okay? They cannot tell. They are on diploids. Okay? Diploids have one from mom and one from dad. But these markers cannot distinguish them. All right? So the first three are useful for looking at similarity and if a band is shared on a gel, it's similar. And you can work out some population differentiation, but you can't measure how a population is evolving. Okay? You can't do a Hardy-Weinberg because you don't have both alleles. You cannot distinguish how many alleles are there in the population. Okay? So if you want to do that, one of these Sanger sequencing, the fourth one, it can be phased. How? We can clone our PCR product. So we want a particular gene from the nuclear genome, okay, the diploid part of the genome. We've amplified it using PCR, we've sequenced it, but it's unphased. We don't know what sequence comes from the mum, what sequence comes from the dad. How do we figure that out with the Sanger sequences of PCR products? We have PCR product. What do? We clone the PCR products into a vector, and then we sequence each clone, and when we sequence enough clones, we will pick up the sequences of each of the two alleles. Okay, so Sanger sequencing can be phased. Sanger sequences can be phased, but you need to go and do cloning. Whole genome sequencing can also be phased. How? If you increase the coverage to above 16x. Okay, if you're unsure about any of that, please watch this part of the lecture again and again and again. You need to know this stuff. You need to know this stuff. Okay? Right. How do we phase a Sanger sequence? I just explained it, but we're going to do it now with pictures. How do we phase a Sanger sequence? By cloning. Okay? So we often see in a Sanger sequence when we amplify a gene from the nuclear part of the diploid part of the genome, from the nucleus, so where chromosomes from mom and dad are both there, we amplify a gene with PCR, and what happens? Both the copy that mom gave and the copy from dad gave, they both amplify at the same time in the same reaction, right? So now we want to see which one comes from mom and which allele comes from dad, all right? Because when you sequence them together, you will get a sequence like this. So let me take you back a few steps. This is the sequence of mom, okay? This is the sequence here, this one here, of dad. And you see the sequence is identical, except for what? Here, in mom, we have a G. And in dad, we have an A. So these are two different alleles. It's the very same sequence, but the G allele mom is giving and the A allele dad is giving. So now if you are the offspring of this mom and dad, and I take your DNA and I do a PCR for this gene, I sequence it with the Sanger method, what will I see? I will see a sequence like this for you, okay? Because at this place here, what will I see? I will see an A and I will see a G. Why? Because mom gave the G and dad gave the A. But in me, they are both in my nuclear genome. The A and the G are in there. The copy from mom and dad are in there. When you do the Sanger sequencing, it amplifies both copies together. So now when I sequence that, what I've amplified in me, or if you are the child of these two, then you will see an A, the black one, and you see this little green one here? That's the G. Sorry, that's the A. The G is the black one. Okay, you will see two peaks. And when you see two peaks, you know, there are two different alleles in this Sanger sequence. All right? So what happens? What happens? I just explained to you why. So what happens when we clone? We take these PCR products, we clone it into a vector like a plasmid, and then we grow up single colonies. And each time we pick a single colony, we know that only one fragment got put into that plasmid. So we're only going to get one of the alleles if we sequence that plasmid that we've cloned. But if we sequence more of them, say 10 of them, then we will pick up, some of them will have the A allele, some of them will have the G allele, and we will know, this individual is a heterozygote because he has an A and a G for that allele, okay? And we have been able to clone apart the two parental alleles. So that is how you can phase Sanger DNA sequences by cloning. This will be a question in an exam or a test. You have to know this. And if you just memorize this, I'm not going to ask you a question in a way that you can just memorize the slide and spew it out to me. No, you're going to fail if you try and do that. Okay? Be warned. All right. So now, let's talk about, we've been talking about diploid markers, phased and unphased, right? Now we have to talk about another kind of marker, a haploid marker. So what is a haploid marker? These markers don't need to be phased. Why? Because the genome that we are looking at has only one allele, okay? They are haploid. So what kind of genome has one allele? A bacteria doesn't have two parents. They don't have sex the way we know it, okay? So the two parents don't combine to give, they're not diploid, they're haploid. They have only got one chromosome, okay? So bacterial DNA is a haploid marker. What else? Mitochondrial DNA, remember we get it only from mom, so it's a haploid marker. Chloroplast DNA, remember we get it only from one parent, and only plants get chloroplast DNA, but it is organelle DNA. Remember, these two were explained by the theory of endosymbiosis. Please, you need to know the theory of endosymbiosis. If you try to buy heart in 2544, you're going to struggle in 3649. I'm warning you, okay? Last year was not for holiday. If you were on holiday last year and you scraped through, you're going to find it difficult this year. All right? Please, understanding, no memorization. What about the Y chromosome? What part of the chromosome is the Y chromosome? The part of the Y chromosome is the part that only males have. Okay? Only males have the Y chromosome DNA. And that part does not have a piece to recombine with on the X chromosome. Okay? And therefore, that part of the Y chromosome is a haploid. You cannot get two alleles. There's only one allele. Bacterial DNA and viruses. Okay? So, in these cases, for all of this kind of DNA, you can just use Sanger sequencing, okay? Or you can use a NGS sequencing at low coverage, okay? Even below 16S, as long as it's above 5x to be reliable. If you know it's a haploid marker, you can go to 10x and you're still fine. Okay, you don't need above 16x whole genomes to get haploid markers. Okay, and these are also because they do not recombine, strictly speaking, they can actually be used. They're very useful haploid markers because they can use to trace what we call lines of descent. They can be used to make a tree or a phylogeny and work out common ancestry. So that's why these haploid markers are actually very useful. Okay, so now let's look at the second way of measuring or genetic variation. We can measure it by the number of copies of a DNA sequence, okay? Variation in copy number. The fact is that some of your genes have more than 2 copies. Why do some genes have more than 2 copies? Because many genes are duplicated, there's been gene conversion, and so on, unequal crossing over. You know how these things work because we discussed them in 2544 during how genetic variation is generated through mutations, gene duplications, gene conversions, and so on. Please, if you've memorized in 2544, you're going to struggle here. All right, so because they've been duplicated, converted, etc., there are now more than 1 copy of the very same gene in your genome. Okay, genes associated with the immune system, MHC genes, HLA genes, immunoglobulin genes, why are they in high copy number in your genome? Why are there so many copies of those? Because remember, you can be infected by many diseases coming in, right? Your immune system has to be ready for any of them. So the more copies of a DNA sequence of these genes you have, the more antibodies, the more combinations of incoming bacteria or pathogens that you're able to fight against. Okay? So the number of copies of these genes is directly related in your immune system to how well you're able to fight those diseases. There are multiple copies of other kinds of DNA. Telomere DNA. For example, you know what telomeres are. Here's a chromosome. This is the piece from mom. This is the piece from dad. Here, they are the homologous chromatids. And what do you have here? You have the centromere of the chromosome, and at the end you have the telomeres. So telomeres are interesting. What are they? They are pieces of DNA with the sequence TTA GGG. Okay? Why do we need? And this TTA GGG is repeated in this blue area millions of times. Why do we have that? You know why? Because in 2544 you learned during the DNA replication, because of the priming, there's always some parts, the end part of the chromosome is always not really replicated. We always lose a tiny bit at the end of the chromosome because of that problem. Go back to your 2544 notes and look at that. You should already know why telomeres get the ends of your chromosomes become shorter and shorter and shorter and shorter every time your cells multiply. Therefore, an old person's telomeres, this blue part here, will be much smaller. They'll have much fewer TTA and GGGs compared to a newborn. Okay? A newborn's telomeres will be very long. So, you can find out, in theory, how old someone is by measuring how many copies of telomeres they have. So you can use a PCR primer and amplify all the telomeres, and then figure out how many copies there are. Okay? So your telomeres always gets fewer as you get older. All right? So it's very interesting to know how many copies we have of certain DNA. Now, how about the number of copies of transcribed DNA? Now, this is extremely useful, especially for diseases. This you can do using RNA-seq or using qPCR. In the old days we used qPCR, nowadays we use RNA-seq. So what can happen? So here we have a normal cell doing its normal job, what it's supposed to do. Normally it produces 2 copies of gene A, none of gene B, and 1 of gene C. But what happens if that cell becomes mutated by ultraviolet, or a mistake during replication, or a chemical has mutated the DNA, and it becomes what? Cancerous. It starts doing its own thing. It doesn't do what it's normally supposed to do. It starts doing it's got its own agenda, self-aggrandizement, like a corrupt government official. It doesn't now work for the body, it works for itself. How do we detect those cells and get rid of them before they become a big cancer and before it kills you? Right? We need to detect it. What happens when that mutation happens? Suddenly, gene A, it used to be 2 copies. No copies are produced. Gene B, where no copies were produced, suddenly gene B is producing 3 copies in this cancerous cell. Gene C is producing another modified copy of itself. So you see, if we now look at this cell, take the RNA, do a transcriptome, and we look at this cell, take RNA, do a transcriptome, we compare the gene expressions, we can tell immediately which patient has got a cancer and which patient does not have a cancer. Imagine how useful it is to know the variation in copy number of RNA. Not just DNA, this is an RNA example. So when I ask you in an exam question or in a test question, why is it useful to know the different copies of a DNA sequence? You better come up with some good answers. Okay? So the last kind of genetic variation, organization of genetic variation that we're going to talk about is the hierarchical organization of genetic variation. Okay? So the hierarchies, and you see here what I mean by hierarchy. So here you got the individual level. In fact, you can look at below the individual level, you can look at the chromosome level. Between the 2 chromosomes inside an individual, what alleles? Here you see they are heterozygote for one big A, small a for one gene, one locus, and for the other locus they are homozygote, big B, big B. Okay? And so that is the individual level. Okay? So you have the individual level down here. Okay? So actually here's the individual level, and this is looking at chromosomes and alleles inside the individual, right? Then you have the population level, okay, within a population, and then between 2 different populations, okay, between 2 different populations, and among 2 or more populations. There are different ways in which the genetic variation can be partitioned, okay, or as we say, structured. So how the variation is structured into these populations is very, very important to know. For example, if we have an endangered species like rhino, like black rhino, we want to say, oh, how much variation do we have in black rhino genetics? Because remember, we want to preserve them for the future, right? So if they have little variation, it means that they're going to be inbreeding soon. It means they're not going to be very healthy genetically. So we need to make sure we measure the variation and we're able to see, okay, the variation is low, let's bring gene flow, let's bring alleles from somewhere else. Or the variation is high, this population is fine. But also we need to see how the variation is structured across the whole species. So now, what does that mean? Black rhino occurs where? South Africa, Namibia, Kenya, and so on. So our black rhino in South Africa, the same genetically as the one in Namibia, or the one in Zimbabwe, or the one in Kenya. They have got their own black rhino populations. So that's what we mean by, is there variation between and among different populations of black rhino, for example. Okay? So where is the variation? Is the variation inside the population? Is the variation between populations? Is the variation at the level of the individual? It depends. Which one you measure depends on what you are trying to find out. As I told you at the start of this lecture, depends on what you are trying to find out. If you're a medical guy looking for cancer, if you're a conservation guy looking to conserve lions or rhinos or elephants, yeah. If you are a evolutionary person looking at how the whole species is structured, it can be for a number of reasons, and depending on your question, you will need to choose one or more of these levels at which you measure the genetic variation. Okay, here we will now look at the individual and population inside a population level, but we will look at the third level between populations when we talk about the next sections in genetic structure. Okay, so the section called genetic structure in this module will be looking at this part here, how the variation is structured between two different populations or among two or more populations. Okay, we'll save that part for the next sections, but we will deal with individual and within population variation here. So individual variation can be what? The number of alleles an individual has, simply the number of copies of a particular gene, the individual heterozygosity, the number of loci at which an individual is heterozygous, divided by the total number of loci that were genotyped. And then what about copy number? How many different copies of your immune genes do you have? Yeah, how many different copies of telomeres do you have at your different stages of life? What different gene expression profiles, transcriptome profiles do you have from messenger RNA in different life stages, in different tissues within the same individual? Okay, you can figure out a lot of things by looking at individual variation. Okay, but you can also find out a lot of things by looking at the population genetic variation, so the genetic variation inside a population. Why is the population important? This, guys, I'm telling you now, is going to be one of the main things you will hear in 3649, and you have to internalize this. You have to make it part of yourself. You cannot not know this. Okay? Why is a population important in genetics and evolution? Why? Why is the population the most important? Why? Because individuals can be affected by evolution. They can undergo selection. They can be determined to be unfit by not making it through environmental conditions. An individual can die or live. An individual can have kids or not have kids. Okay? So evolution works at the level of the individual. But an individual cannot evolve. Okay? You know this from 2544. An individual cannot evolve. An individual can be born. An individual can survive the struggle if he's good enough, or he or she is good enough. They can survive the struggle to reproduce. They can have kids of their own. And then what? They can die. An individual can't do anything more. You are not going to, there's no heaven or hell. You're not going to any afterlife. You die. End of story. Okay? You don't evolve. You don't evolve. Because if you could evolve, it would be like what? Inheriting acquired characteristics. Whatever you acquire in your life, if your hand gets chopped off, if you get cancer, and so on, you can't give that to the next generation. Okay? So individual cannot evolve. Only what can evolve? The population. Okay? Only the population can evolve. Okay? So when we want to study evolution, we must look at the variation of a population. And we look at that and we say, is the variation changing? Is it high? Is it low? Is the variation changing from one generation to the next? That is how we measure evolution, by looking at the population. We don't measure evolution by looking at individuals. Okay? That's why population genetic variation is so important. And that's why there's so many ways to measure it. You can measure it looking, for example, when you're looking at microsatellites, you can measure the mean number of alleles. And guys, it was okay last year to give MNA and expect to get a mark in a test. This year, it's not like that. You're going to have to tell me what MNA means. What does the mean number of alleles means? It means what? Taking all the alleles in the population, okay, and dividing it by what? The number of loci, okay? So you need to know how we calculate the mean number of alleles, not just by hearting mean number of alleles. You're going to fail if you try and do that, okay? When we correct the mean number of alleles for different sample sizes, it's called allelic richness. Okay, we usually correct using a bootstrap or a jackknife or a rarefaction approach, but it's allelic richness is the same as mean number of alleles, except if we have a population where we looked at 5 individuals and we have a population where we looked at 100 individuals, you can't compare the mean number of alleles between those two populations, but you can compare the allelic richness, okay, to see which population has a higher or lower diversity, okay, because allelic richness is mean number of alleles that is corrected for samples of different sizes, okay. So please, no by hearting this year. Heterozygosity observed and expected, you already know that you need a phase marker for sure, and this is the average number of observed heterozygotes. So basically, look at all the genotypes in the population, how many heterozygotes are there, that's your observed heterozygosity. Expected heterozygosity is not the same. Expected heterozygosity you can work out from what? You work out from the allele frequencies. And expected according to what? When you say expected, it means there's something you are expecting. Why would you expect something? Because something has happened, therefore you expect something, okay? So what has happened and then you expect a certain heterozygosity. So Hardy-Weinberg is what has happened. Expected heterozygosity basically means if this population was in Hardy-Weinberg equilibrium. In other words, if no evolution is happening in this population, what heterozygosity can we expect? That is what expected heterozygosity is. Not something to do by heart, something to understand. Okay? With an unphased marker, or with a haploid marker, you can figure out the number of polymorphic sites. Okay? Like in a Sanger DNA sequence. You can line up the different sequences in a sequence alignment and figure out how many polymorphic sites there are. That is a way to measure the genetic variation in that population in your alignment. You can work out from the same alignment what is the average number of nucleotide substitution per length of DNA fragment. That's the nucleotide diversity. You can work out the haplotype or gene diversity, which is basically how unique is a particular haplotype in a given population. Basically a particular allele. Okay? Haplotype is basically what? It's another way to say allele. And when do we use the word haplotype? And when do we use the word allele? Yep, the clever ones of you have figured it out. When you have a diploid phased marker, you refer to alleles. When you have a haploid marker, you refer to haplotypes. Okay, that's the difference. Okay, and lastly, there's Watterson's theta, which we'll get into much later on in the module. It is basically a way of quantifying the variation in DNA sequences, and you do that by multiplying the effective population size by mu or u, this parameter here, which is the mutation rate times the effective population size multiplied by 4, because you're getting 4 gametes from each parent. There are 4 gametes that are segregating in each parent. You are taking drift into account with effective population size, and you are taking the mutation rate. So 2 of the main forces of evolution are being taken into account in Watterson's theta, another way to measure population genetic variation. So I think I'm going to leave it there, okay? This was quite an intense lecture. There's a lot of information in this lecture. You will need to watch it again and again and again in order to answer the test and exams questions that are coming from this lecture. It was a lecture that is going to feature a lot. So please watch it again and again, and I will find you. That concludes our lecture on variation, lecture set on variation, the first section. So we'll now be going on to the section, and I look forward to seeing you all there.